So this is a GCSE presentation looking at the Unit B2 in the AQA specification, which is organisation. In particular, we're going to be looking at the eukaryotic cells or plant and animal cells. So the first task is to go back over your notes from Key Stage 3 and see if you can remember any parts of any cell. And if you do, you should be able to identify any part of a plant and animal cell. And plants and animal cells collectively are known as eukaryotic cell. You will go back and you will go through other presentations and they will take you through a different group of cells which are known as the prokaryotic cells. But as I said, we'll cover them in a different presentation. So first question, what can you remember from Key Stage 3 in terms of things that you find inside plant and animal cells? So by the end of this presentation, you should be able to go and label a diagram of a plant and animal cell. Remember, they're called the eukaryotic cells. You should be able to identify the function of each part, the function of the organelles inside each of those plant and animal cells. You should also be able to identify the different levels of organization and examples of different structures that are found at each level of organization. Now, at this point, you should have a list of components which are called organelles, or parts of a plant and animal cell. In front of you, you've got a picture, or sorry, a diagram of two different shapes of cells. One's more square in shape and one is more circular in shape. And what I'd like you to do is pause the video and identify which of the two cells that you're looking at represents an animal cell and which of the two cells in front of you is representing a plant cell. So pause the video now. Which one's plant, which one's animal cell? Now, if you've made the classic mistake, you have said the one on the left, the square rectangular shaped one, that is a plant cell, and the one on the right is an animal cell. Now, if you have said that, that is incorrect. The truth is the one on the left hand side is an animal cell, the one on the right hand side is a plant cell. The reason we identify them as animal cells and plant cells is not because of their shape, but because of what things we find inside each of those cells. So it's the structures that we find that identifies if it's a plant or animal cell. Do not make assumptions about the shape of the cell automatically means it has to be a certain type. The only way to identify what type of cell you're looking at is by what you find inside it. So the one on the left is an animal cell, the one on the right is a plant cell, even though they're not the shapes that you're used to. And when it comes to drawing and labeling cells, there's a couple of rules, and one of those rules is flexible in exam conditions. So you should really, when labeling any diagram in biology, use horizontal lines. They should always go left or right and should always be straight. They shouldn't go at an angle. However, if you did that in an exam, it wouldn't be a problem as long as you draw a line with the ruler. It has to be a straight line. You have to touch whatever it is that you're labeling. You must make sure that line makes complete contact with whatever it is you're labeling. And there must be no arrowheads. Do not put any arrows. It's just a straight line. At one end, you touch the thing you want to name, and at the other end, you have the word that you want to use to identify that particular structure. Best practice is horizontal lines, but you'll not be penalized if you drew a line at an angle or vertical line in the exam, as long as you labeled it correctly with the ruler, touched the object you're labeling, and did not include arrowheads. So we're going to start by identifying each of the parts of an animal cell. And while the animal cell shape will change, these structures will not. The one I'm hoping you would have remembered from Key Stage 3 is the nucleus. The nucleus, you may know something about it, controls what happens in a cell. Please don't use that at GCSE. At GCSE, you should be saying that the nucleus stores genetic information or that the nucleus stores DNA. And if you look on the diagram, the nucleus is always the large dark patch inside the cell membrane, which would mean that that is labeled the nucleus. The cytoplasm is the liquid that all of the material inside the cell floats within. It's also the place where most of the chemical reactions happen, so that's what you're expected to memorize as the function of the cytoplasm, and we can label anywhere in that diagram that is basically white and not inside another organelle. The cell membrane is the layer around the outside. I always like to think of a cell membrane as kind of like a water balloon. It's the actual balloon, whereas the water inside would be the cytoplasm. So the cell membrane we take as any of the thin layer around the outside, and we can put that down as a cell membrane, and its function is it controls what moves into and moves out of the cell. So it controls what flows into and flows out of the cell is its primary function. The next thing we look at is the mitochondria, and the mitochondria are, the best way of describing the appearance of a mitochondria is think of a hot dog with mustard or ketchup squirted over the top. That's the best way of describing the appearance. When you get to A level, you'll find out a lot more about its appearance, but for GCSE, that's all you need to be able to do is identify what it looks like on a diagram. And its function is aerobic respiration. Now, if you don't know what aerobic respiration is at this point, don't worry. We will go through that in another lesson in much more detail. But respiration is not breathing. I know you may have been told that in the past, but please don't say that anymore. It is not breathing. Respiration is a chemical process that results in the release of energy. We will go through it in a lot more detail, folks, so don't worry about it for now, other than mitochondria is the place where aerobic respiration occurs. And lastly, we have the little black dots, and they're called ribosomes, and their job is to make proteins. So now we're going to look at a plant cell. Now, this plant cell is a bit different to a normal plant cell diagram that you'd be used to seeing, and I'll explain why a little later on in the presentation as to what the big difference is, but for now you'll just have to trust me on this one. On the right hand side of the picture you have the same 5 structures that were identified in the animal cell. So we still have a nucleus, we still have the cytoplasm. The cell membrane is the layer that contains, it's the water balloon, remember, it's the actual balloon, and it contains all of the cytoplasm and all the organelles inside it. We have the mitochondria, which again, in terms of what they would look like in a diagram, look for that hot dog with a mustard or ketchup squirted over the top. And then the ribosomes are the little black dots. In plant cells, we have 3 additional things we need to be able to identify. The first thing we need to be able to identify is a thing called a chloroplast. And a chloroplast is simply a circular shape, and normally if it's in color you would see it as green in appearance. And the reason it's green is because it contains chlorophyll. And chlorophyll is a chemical that absorbs sunlight energy. The next thing we've got is we have a permanent vacuole, and the permanent vacuole is a large store of fluid in the form of cell sap. And the last structure you find in a plant cell is called a cell wall. And the cell wall is a really thick and rigid layer that goes around the outside of the cell membrane, so it surrounds the cell membrane, and its purpose is for strength and support. Again, folks, you need to know the names of each part, you need to be able to spot them in diagrams, and you need to be able to go through the functions of each of those parts. Notice the 3 on the left, chloroplast, permanent vacuole, and cell wall, are the ones that are only found in organelles. And if you go back to the original bit of the presentation where you had the 2 slides side by side, you'll see that's how you were identifying which was the animal cell and which one was the plant cell. Now, previously, you will have seen plant cells and they will have looked a little bit more like the picture on the left rather than the picture on the right, where you have a cell wall and then the inside line of the cell wall you've identified as the cell membrane. Well, we've got a reason for that. If you look in the picture on the left and right, what are the differences between those 2 pictures? Pause the video, have a look, see if you can identify what is the difference in the 2 pictures. If you see on the picture on the left hand side, the cell membrane is actually touching the inside of the cell wall. On the right hand side picture, the cell membrane has moved away from the cell wall. It is smaller. Now the reason for that is because if the permanent vacuole, remember that's the one that contains the cell sap, is filled, then the cell membrane will swell up as much as it can. The water balloon will get as big as it can. But if you imagine that the water balloon is inside a giant box, and the box is the cell wall stops that cell membrane getting any bigger. It can only get as big as the box. The water balloon will fill up to the box and it won't get any bigger. So the cell membrane is pushed up tight against the cell wall, which is why we can't actually see a separate cell membrane on the picture on the left hand side, because it's pushed up tight touching against the cell wall. On the right hand side, the permanent vacuole is not filled. It has less water, it's not a capacity. So the water balloon has shriveled up, the cell membrane is shriveled up, and the cell membrane has pulled away from the cell wall. Now, rather than explaining those concepts in an exam, we can simply say that if the cell is full of cell sap, the vacuole is filled, and the cell membrane is pushing up against the cell wall, we can say that the plant cell is turgid. If, however, we get a cell membrane and the cell membrane has pulled away from the cell wall because the permanent vacuole is not filled, we refer to the cell as being flaccid. So we can use the terms turgid and flaccid to describe plant cells where the cell membrane is touching the cell wall or the cell membrane has moved away from the cell wall. One of the things they may ask you to do in the exam is to put organelles found inside the cell into order of size. And the four they'll ask you to look at is the nucleus, the ribosome, the chloroplast, and the mitochondria. Pause the video for a moment, have a go at which one's the biggest, all the way to which one's the smallest, put them in order of size, and then restart the presentation. The largest, we start with the nucleus. Smaller than the nucleus is the chloroplast. The mitochondria are a little smaller than that, and by far the smallest is the ribosomes. You need to know that sequence going from largest to smallest organelle. It'll also help you when you're trying to identify which bit's which inside the plant and animal cell. Remember, plant and animal cells collectively are called the eukaryotic cells. So what we've got now is what we call the levels of organization in living things. And it's basically if you get any living organism and subdivide it down into individual components with the different levels that we can break it down to. For example, we could have a house which can break down into rooms, which we can then break down into individual walls, which we can then break down into individual bricks. Well, the building blocks or building bricks of any living organism are known as cells. They're the smallest living unit in any organism. If we get a group of cells and they all carry out the same function, we refer to that as a tissue. Now, tissues each have their own specific function, but if we get a group of tissues and they all work together to carry out a particular job, we can call that an organ. We can also get groups of organs working together, and groups of organs working together gives us an organ system. And of course, organ systems can then be put together to form a particular organism. So you need to know the definition of each of the levels of organization in living things, and you also need to be able to describe each one with a definition. So some organisms are made up of just one cell, and they're called unicellular organisms. Levels of organization only comes into what we call multicellular organisms. So basically anything beyond a bacteria or algae or any of those kind of very small single cell organisms, anything beyond that living creature will be a multicellular organism, which means it will have cells, tissues, organs, organ systems. Most importantly, in the exam, they quite like to give you plants as an example because everybody knows all about the body in animals because it's our own body, but people are much less familiar with plants. So just be aware that plants have different systems just like in all living animals. They have organs, for example, a stem or a leaf or flower. They have individual tissues within each of those organs, and of course every one of those tissues can be made up of different cells. So in this presentation, there's a lot of terminology. What I'd suggest you do is get yourself a big piece of paper and start writing down all the words that you can connect to this particular presentation and build yourself into a giant web with the lines between each of the words that you can remember. Put a little explanation on how those words are connected. Once you've done that, everything you can remember, go back through the presentation again, change color pen and add in other words or other examples from textbooks, websites, or any other sources of information so that you've got the ones that you remember to start with and the ones you need to memorize to make sure you're ready for the exam.